one as well. Well, thank you very much for that introduction, and thank you all for coming along to hear about my life in slime. I can't actually recall when I first saw a slime mould, but I definitely remember seeing this one on King Island, not exactly this one, a species like this on King Island when I was surveying birds there about 20 years ago. And I touched it and a lot of spores dispersed, and I had no idea what it was, but I consulted the local mycologist and she told me it was a slime mould. Sorry, I have got a mic here. You can hear me? You can hear me? Sorry, I'll speak up. My first proper introduction to slime moulds was through the Fungi Mat Project. The Fungi Mat Project is a citizen science project initiated by the Victorian Field Nats and the Herbarium in Victoria. And it was a project, well it still is a project, to document the fungal species that occur in Australia. They started off with 5 what they called target species. So these were species of fungi that were easy to identify in the field, either from photographs or because they were illustrated in field guides. And they gradually built up the number of fungi that they wanted people to search for, and eventually they added slime moulds to the mix. Not that slime moulds are fungi. So the 3 possibly most commonly seen slime moulds, and certainly not the most common, but the most commonly seen, are these ones, and they're quite conspicuous. The one on the left is a very common species that you'll find on very wet wood or wet bark, so it's been common this year. The one top right, Fuligo septica, also known as dog's vomit slime mould, and that's one that you might come across in your garden. It occurs on mulch. If you're out walking in the bush in the summer, you often see it on logs and stumps. So it is a common species. And the bottom right one, Lycogala epidendrum, or actually possibly not epidendrum, because at the moment there's a huge revision of taxonomy, and a lot of the species who we thought we know are actually proving to be different species. My first encounter with slime moulds involved looking at the associations with invertebrates. So I've always been interested in what species occurs on what plant or tree, and in this case I was finding a lot of Collembola, also known as springtails, feeding on the developing slime moulds. I contacted the local springtail expert, Dr. Penny Greenslade. She didn't know that springtails fed on slime moulds. Even the leading springtail expert in France didn't know they fed on slime moulds. But a lot of people who study myxomycetes or slime moulds did know they fed on slime moulds, because they tend to be a bit of a nuisance because they feed on them. Anyway, the species bottom left is an Acanthanura species. It's an undescribed Collembola that's only found in northern Tasmania. I think it was found at the gorge, and it's been found at our property. And that species and a whole range of similar looking Collembola feed exclusively on slime moulds. The first species that I kept finding in 2010 when I started looking for them in the forest around home was this species called Eleomyxa cerifera. I had no idea what it was. I didn't have any field guides at the time, and the only way I could find out about this was to send a photograph to a friend in Victoria, Paul George, secretary at the time of Fungi Mat. It took a while to actually convince him it was a slime mould, because it was often on mosses, and sometimes it looks like just an extension of the moss. But I convinced Paul that it was a slime mould. He eventually tracked it down to this species. The book that he was referring to stated that it was a very rare species, and the yellow waxy collar was only known from Japanese collections. My record of this was the first record for Tasmania. There was one other record at the time for New South Wales. And it hasn't been as common since at my study site, but a lot of people have found it elsewhere in Tasmania and in the wetter parts of southern Australia. Another one of my first finds was this species. I was photographing species in the field at the time. I eventually got around to collecting this species and sending it to the herbarium in Melbourne. I didn't know what it was. I actually did. I misidentified it immediately as Tubifera bombarda, a species known only in the tropics. And because it seemed a bit doubtful that a tropical species would turn up in Tasmania, as we know, Tasmania is far from tropical, I wrote a little article for the Fungi Mat newsletter, and as a result of that, Dr. Steve Stevenson, a researcher in the US, accessed my collection from the herbarium in Victoria, and he accessed my private collections. And at the time, a researcher, Dr. Dmitry Leontev, was studying this particular family, the Reticulaceae family. They could actually tell just on looking at it that it wasn't what I thought it was, Tubifera bombarda, but a species new to science, and they called it our Wysioloidia. What was interesting about this was that Dmitry Leontev thought that it is the ancestral stock of this family, this Reticulaceae family, and because Australia and Tasmania are well-known refuges of relict biota, which means that the species that were once very common but have now restricted, very, very restricted distributions. This one has been found in Victoria, New South Wales. In fact, somebody found it in Queensland fairly recently. So it's not all that rare. It's just that not many people are looking for slime moulds. Whoops, wait, sorry. This is another one that I found the first year I was photographing slime moulds. And it has turned out to be very exciting because it looks like it's a new genus of slime moulds. Now, new species of slime moulds turn up fairly regularly, but new genera much more rarely. I, once again, very confidently misidentified this species. Several collections of this, plus some other species, eventually made their way to Dr. Gabriel Moreno in Spain. He didn't know what it was, and I have since sent him a type specimen. Unfortunately, that type specimen didn't have well-developed spores, so I've just recently, in fact in the last month, sent him another type specimen. The type specimen of a species is the collection on which the original description of that species is based. So since the other exciting thing about this is somebody, as I was collecting the type specimen in Tasmania in July, somebody in Victoria had also found the species in Victoria. And actually it's quite widespread in Victoria. It's very easy to identify when it's young. So we know there are quite a few records of this species. Peter MacDonald, who found this in Victoria, put her photographs of the fruiting body, the spores, and other features on the slime mould Facebook page. Now, I'm not a Facebook user, but Dmitry Leontev, the aforementioned, is, and he was very excited when he saw this species because he couldn't place it. He didn't know what genus it was, and they think it is a new genus. As a result of that, he sent me some Eppendorf tubes. They're those little tube things down there, and he asked me to put 100 spores of all my collections in the tubes. Now, this was a bit nerve-wracking. It would have been quite easy if he'd said, just put a heap of spores, as many as you can fit in these tubes, but only 100 was a bit more difficult because you actually can't see what you're doing. You have to do it all under the microscope. So I hope I've done it correctly. They're on their way to Ukraine, but because of the unmentionable virus, it could take a while before the species is, we figure out what it actually is. But Dimitri will use the spores that I've collected to try to extract the DNA, and once he's done that, he'll be able to figure out where it fits in the scheme of things. And because it's a new species, I've been documenting everything I know about it. I found it on that log, and I documented the early stage, the more mature stage in the field, and I have been taking photographs of species with a more powerful camera, and also taking photos of what's inside the spore mass. So these threads are called capillicium, and obviously the spores as well are very important in the identification of slime moulds. Another species that made its way to Dr. Moreno in Spain was this Lamproderma. Another species I was unable to identify. By the time I sent this to him, I did have a few field guides, but even though the species is reasonably common, it wasn't in any field guides, at least that I could recognise. He identified it as Lamproderma acuminulatum. The type locality for this species is Tasmania, and it was described by the Reverend Berkeley in 1839. And it was probably collected, is that right? 1859. It was probably collected either by Ronald Campbell Gunn, who, as people here possibly know, was a very avid collector. He was sending a lot of plant material and a couple of slime moulds to Kew Gardens, to Hooker, and it was included in the first Flora Tasmaniae by Hooker. Gabriel Moreno also sent me what he called a colour plate. He said, I'll do a colour plate for you. And so a colour plate involves just photographs of the fruiting body, what's inside the fruiting body, the spores, and then he has access to a scanning electron microscope. And so those spores along the bottom there are micrographs taken with a scanning electron microscope. Unfortunately, I haven't got one of them yet. They're quite expensive, I believe. But it was a good lesson. One of the other species Gabriel Moreno was able to identify for me was this Eleomyxa reticulospora. He sent me a paper describing this species. It was described, another one described as very rare, only known from the type locality, which is Java, and only grows on moss. I found it so many times where I live. It is the most common species I find. I've also found it at Liffey, the Minnow Creek near Sheffield, and it doesn't grow on moss. It's a very common littered dwelling species. So this just really demonstrates that not many people are looking for slime moulds, although it's certainly increasing lately. But because Dr. Moreno had sent me this colour plate, I realised that that was a really good way of trying to figure out what the species are. So I started doing a colour plate of all the species I collected, and that just involves the one on the right, just a description of all the features of the fruiting body, where it's found, and photographs of all the relevant microscopic details. And then there were 3 new tubiferas. Tubifera is another quite large genus known throughout the world. A lot of them, one particular species, Tubifera ferruginosa, is bright red when it first appears. And the colour of this early stage can be a clue to their identification. I found 3 species I was unable, or 3 different tubiferas, well I thought they were, that I was unable to identify. Once again I wrote a post for my website. I sent the link to Dr. Steve Stevenson in the US, who eventually sent it to Dmitry Leontiev, and he, based on, once again, the DNA mostly, was able to establish that there were 3 new species of slime moulds. So that resulted in a paper, 3 new species of tubifera from Tasmania and New South Wales, which I was the senior author. So far, I think they've only been found in Tasmania, apart from the one Tubifera vanderhulliae up on the top right. I don't think they've been found elsewhere. They may have been, but it's hard to know unless you see all the different stages as the fruiting bodies are maturing in the field. So now we come to, what are they? This is a photo to give you some idea of the scale of slime moulds. This is a little species that I found on a gum nut, and the gum nut's about 10 millimetres diameter, so you can see the slime moulds they're about 1 to 2 millimetres high, and that's the average size of the ones that I'm collecting. Obviously the tubiferas and some of the others are quite a lot bigger, but average size about 2 millimetres. And they come in a beautiful range of colours and forms, quite exquisite for something so small and unnoticed. And they have confused taxonomists for centuries. In about 1750, when Carl Linnaeus, who was known as the father of taxonomy, was trying to figure out and name all species of plants and animals, he decided that there were two kingdoms, the plant kingdom and the animal kingdom. At the time, fungi weren't really considered much. They were thought to be plants, so they went into the plant kingdom, and slime moulds, which were at the time thought to be fungi, also went into the plant kingdom. Not long after that, with the advent of the microscope, or the improvement to the microscope, the fungi kingdom was added and slime moulds went into the fungi kingdom. And the reason they've often been described as fungus-like organisms and have been confused with fungi is because they reproduce by spores. They're often found in dark, wet places in a similar situation as fungi, and they're traditionally studied by mycologists. However, they have a few other stages. They have a moving feeding stage called a plasmodial stage. And based on that stage, they were put briefly into the animal kingdom, because the only other things that move about and feed, known about at the time, were animals. So they had a brief period in the animal kingdom, but then, not long after that, their single-cell amoeboid stage was discovered. So this is a single-cell amoeba. It lives in the soil. It feeds mainly on bacteria. And based on this stage, they're now classified as amoebozoans in the kingdom Protista or Protoctista. The Plasmodium certainly are fascinating organisms to watch. So these move about and feed. These were some of my earliest photographs. This was on a standing dead small Banksia, and I think the photographs were taken a day apart. So the change in colour from yellow to green might be because it's been feeding on algae, and that gives the Plasmodium some colour. But they also, some of the species feed on fungi. So on the left-hand photo, actually in both photos, you see that pink, what's known as a resupinate fungus, and the slime mould, I think, was feeding on that. I never actually saw associated fruiting bodies with that Plasmodia, but I suspect it was this species, which is a very common species, Physarum virida. It's actually very common on our woodpile through the year, which doesn't say much about the dryness of our woodpile, in fact, the wetness of our woodpile. Another really interesting Plasmodia is this one. It's a fungivore, so it feeds on the surface of logs, and this one was feeding on this toothed fungus. And I've been watching one of these for the last couple of months, feeding on a white fungus just on the surface of the log. And eventually, for no apparent reason, we don't really know what triggers them to form fruiting bodies, it will eventually form these beautiful fruiting bodies that will later go darker as they fully mature and the spores dry out. There are a couple of different types of Plasmodia. Another one is called an Afenoplasmodia, Afeno simply means invisible, and these Plasmodia are usually active within the substrate, so within large logs or in the soil, and they very rarely come to the surface. They sometimes do, obviously, this one did, and when they form fruiting bodies, they look something like this. So this is actually an early form of the slime mould I showed you first. So this one will eventually go brown and the spores will dry out, and hopefully you'll be able to identify it, but they are difficult. Another little species, this one arises from a protoplasmodia. Proto simply means first. These plasmodia are microscopic, and this fruiting body is about 1 millimetre high. I'll briefly describe the life cycle, and it says it's a typical life cycle, but this is probably based on a species that's very easy to culture in the laboratory. I think the life cycle of species in the field is probably very different, but how do you know? Because most of the stages are invisible to the naked eye. And this is a typical life cycle of an acellular slime mould. There are several different types of slime moulds, and this often leads to some confusion. There's a group called the cellular slime moulds. They're microscopic. They function completely within the soil, and you don't actually see them unless you've got a microscope. So what I'm studying is the acellular slime moulds, or the myxomycetes. And when I say acellular, the plasmodia, the photos that I showed you before, that's one giant cell. It doesn't have cell walls within it. It's just a mass of nuclei surrounded by a cell membrane. So I start with the fruiting bodies at the top. That's what I'm collecting and studying. And from them come the spores. And from the spores come the amoeba. And the amoeba actually feed on bacteria in the soil. And what's been realised fairly recently is that they're actually very important in the soil, because they feed on bacteria, and bacteria has more nitrogen in it than the slime mould amoeba require. So they put nitrogen back into the soil, and they're very important in mediating the flow of nitrogen through the soil and up through the food web. They multiply by simple cell division. They eventually form a zygote, and then they form the plasmodium, and from the plasmodium come the fruiting bodies. They have a couple of dormant stages. So at the Amoeboid stage, they can form dormant structures called microcysts, and at the plasmodial stage, they can form dormant structures called sclerotia. And these form when conditions are unfavourable. So if they run out of food, if it gets too dry or too wet, or any other reason that slime mould amoeba might not like, they will form these dormant structures, and when conditions are favourable again, they will reactivate, they will take in more moisture, and they will start moving again and feeding. So there are about 1,200 species of slime mould known worldwide. I think last time I gave a talk there were about 1,000 known, so new species are turning up quite regularly, and that's mainly because they're one of the least studied of all the microorganisms at the fruiting body stage. So at the fruiting body stage that I'm studying, they're not widely known. At the plasmodial stage, they're one of the most studied of the microorganisms, and that's because the plasmodium of one or two particular species are very easy to grow in the laboratory, and they apparently are very fond of rolled oats. Australia has more than 330 species, and that would be because we've got such a wide range of habitats, so from the tropics to Tasmania and down to sub-Antarctic Macquarie Island, where new species have been found by Dr. Steve Stevenson. So there were 42 species known before my study began, and I have collected at least 120 species. This is really hard to establish because a lot of them are very difficult to identify, so there might be a lot more. They're also very irregular, so you don't see them in the field the same place year after year. So there might be a lot more species. More work will determine this. And the question I ask myself and other people ask, is it a particularly rich site where I live? So I live with my partner Ron at Black Sugarloaf, which is at Birralee. This is an aerial photograph of our place. As you can see, it's completely surrounded by forest, very nerve-wracking in the summer months, but nevertheless, we quite like living there. The temperate forests of the world are known to be the richest in slime moulds, and that's probably because of the constant moisture that they like. But there are numerous different substrates, different plants that I've noticed they are particularly associated with, and I'll talk about them. Also, Tasmania's climate is very favourable to slime moulds. They like wet weather, so the last couple of weeks there's been a lot of activity in the field. They'll respond quite quickly to moist conditions. So a couple of days after rain, I generally go out and check for slime mould. Actually, I go out every day, whether it's rained or not. And obviously, daily access to a study site is an advantage. When you're studying organisms that are ephemeral, they don't last long in the field, completely unpredictable, and very small. And one of the reasons being in the field all the time is good for studying slime moulds is a lot of them start very brightly coloured. So this was, I found it a couple of days ago in the forest, very bright coloured species called Trichia discipiens. Once it's mature, it'll be about the same colour as the log. They're very difficult to see. So it's certainly an advantage to see them at this early stage. And I have quite a lot of coloured ribbons through the forest, or pegs, because I learnt very, very early on that if I'd seen one this colour and went back a couple of days later and couldn't find it, it was because they're just almost invisible. So I do mark the spots where I find most of these species now. And some of the particular substrates in the forest are really rich in slime moulds. So our property would have been logged in the 60s, 50s. There are a lot of big old fallen eucalypt logs now covered in lots of leafy liverworts and lichens, and they can be really rich for slime moulds. In fact, that log, the top one, right at the moment, there's this species, beautiful Lamproderma gracile, that I've only just found out the name of. It used to be a subspecies of another Lamproderma, but it's now been given species in its own right. Another one that I found on the log was this one. This is our Wysioloidia again. It's slightly younger than the version I showed you before. And you notice those little pin-shaped structures on the fruiting bodies that are slightly bent over in the middle. They are spermatophores, and they're produced by collembola, the springtails that were feeding on the slime moulds right at the beginning. So the male deposits them on, not necessarily on fruiting bodies of slime moulds, but through the wet areas on the lichens, and the females come along, and that's how they multiply. This is another species that I just found on the same log, so that's another Trichia species. And sometimes that log that I just showed you has a lot of different species, and other times it won't have any species, And one year it had this species, which is Cribraria cancellata, a really exquisite little slime mould. And it had a whole range, it had about 5 different Cribraria species on that log, and I haven't seen them there since. So that's another advantage of being in the field. They're really irregular. You don't know when they're going to turn up. They might be very common one year, and then you won't see them for years. So it's certainly an advantage to be in the field all the time. Another really rich substrate is, I find, in the tree ferns. And this tree fern was a really good source of slime moulds until a couple of weeks ago, when it was really windy at home and a whole lot of the dogwood, which are those tall, thin trees in the photo on the right, blew over and blew right on top of the tree fern on the left. But it was actually really interesting, because what happens is, over the years, leaves, sticks, bark, big limbs fall onto the tree ferns, and I was able to just tease apart some of the material. It's just like soil in the tree fern, and they can be really rich in slime moulds. So that was a fun and quite an accidental discovery. But I have found species there that I haven't found in any other areas. And they're mainly litter-dwelling species. They probably occur in the leaf litter that's scattered on the ground, but it's very convenient to look in a Dicksonia, a tree fern that's about my height, because I can tell you, crawling around on the ground looking for slime moulds is not good for the back. This is another species that I found in the tree fern, and it seems like it's another species that's new to science. I'd only ever found it in 2 tree ferns at home, and they were adjoining tree ferns. Now, sometimes one tree fern will have a lot of species in it, and the adjoining tree fern won't have any. But one year I found it in one tree fern, the next year I found it in another tree fern. They were about just a couple of metres apart. And one of the prerequisites for describing new species is that they must be at least 50 metres apart. So I didn't really think much more about it until I was checking the iNaturalist website. If people aren't familiar with iNaturalist, it's a website a bit like, I suppose it's a bit like Facebook or Instagram in a way, in that you can post photos there, but it is a scientific website. So you can post photos of birds and flowers and slime moulds, fungi, anything. And somebody, actually Brenda Marshall from Loongana, had found this species in tree fern at Loongana. And that, I think it's probably a bit closer than 100 metres. There is a more direct route than Google showed me on the map that I downloaded, but certainly more than 50 metres apart. And because of that, both collections, both my collection and Brenda's collection, they're now in Spain with another researcher, Dr. Carlos Lado, and we're yet to hear back the outcome of that investigation. Leaf litter abounds in the forest. It's just, the forest where we live is pretty much untouched. We can't actually get there. I mean, you could if you had a big bulldozer, but it's just walking tracks through the forest. So there's heaps of litter, there's lots of fallen logs, things are falling all the time, and it never gets tidied up. So this is one of the litter-dwelling species, absolutely exquisite, Criterium minutum. This is another one growing on dogwood leaves. If people are familiar with dogwood or Pomaderus apetala, you'll know that they've got very hairy leaves. You can see the stellate hairs on the bottom of the photo, and they seem to be particularly favourable for slime moulds as well. This is another litter-dwelling species. Once again, really brightly coloured when it first appears. It's just a matter of being at the right place at the right time to see this sort of species, because if I hadn't have seen it at the yellow stage, I wouldn't have had a chance of seeing it. It just looks like a grey, I mean, you can see there it has got structure in it, but in the field you wouldn't really pick it unless you'd seen that yellow bright stage. Another really rich substrate is the native Clematis. And at home, and I actually haven't seen it this size anywhere else, but at home the base of that Clematis, and that's that one curling around there, is about 20 millimetre, was it 20 centimetres at the base. It climbs right up to the top of the blackwood trees. But when the stems die, the bark is very absorbent, and it's fantastic for slime moulds. And once again, I've found species on the Clematis that I haven't found anywhere else. They're not particularly, I thought the one bottom right was another undescribed species, but I think it's a described one. They're not particularly uncommon, it's just interesting that they occur on the Clematis, and I haven't seen them on any other substrate. And another plant, Bedfordia, so blanket leaf, Bedfordia salicina, is a small understory tree in the Asteraceae family. Once again, it has similar bark to the Clematis, very absorbent, so when the trees die, and I tend to find more on dead trees than living trees, once again, very rich substrate for slime moulds. And the dogwood is also very rich for slime moulds, so dogwood, Pomaderus apetala. One thing I found really interesting is what I call hotspot logs. So these logs, there might be, I think in that bottom left photograph, just one of those logs had about 10 different species over weeks and months a couple of years ago, and the adjoining logs had nothing on them. And this is something I find every year. So every year I try and seek out the hotspot log and see if I can just monitor the species as they're growing and as they're maturing on the log. So there are some compounds in slime moulds that are important, but whether they'll ever be able to be used is doubtful, or is a problem, because most slime moulds are really small and you can't get enough material from the slime mould to be able to use it effectively. This one has been used, or could be used, in the bioremediation of soils. And this little one here, the Asteria, has anti-cancer compounds. And there are other slime moulds that have anti-Parkinson's disease compounds. So it'd be interesting to see where the research goes with these compounds in the myxomycetes. And this is, on the left, I have now, after several years, built up a collection of field guides. I've got microscopes on, I've got cameras on microscopes, and I have a camera for field photography, and the cabinets on the right have my collection of over 2,000 matchboxfuls, representing, as I said, 120 different species. I do send duplicates of most of my collections to the herbarium in Melbourne, at the National Herbarium in Victoria, housed at the Royal Botanic Gardens, and any researcher associated with an institution can access those collections. And this is one of the first depictions of slime moulds by Ernst Haeckel. I've got it on my t-shirt, and just exquisite work by Haeckel. He has a whole series of work of different organisms. So he does marine organisms, plants, animals, all sorts of things. Beautiful work. And if you have like to know more, I've written a book, I have a website, and I'm happy to answer your questions.
Thank you. When you ask your questions, would you wait for the microphone to come to you and avoid touching it in any way, because it's extremely sensitive? just to start off, people of my age were upset when the fungi were evicted from the plant kingdom because we can no longer play animal, vegetable and mineral.
Oh, right.
How do you define a kingdom these days?
That's a tricky one. I think you'd have to ask a taxonomist. But possibly since I think there are a lot more kingdoms now than just plants, animals, fungi and protisters. Yeah, it depends on the taxonomist and how they approach things. But I think taxonomists like to keep themselves in work, so they're always changing. Sorry if there are any taxonomists in the room.
Through the life stage of the various beasties, a colour change occurs. Is there any evolutionary function for that you know of?
I have no idea about the colour change and why it occurs and what makes it occur. One of the really interesting things about slime moulds is a lot of them are iridescent, and that's hard. I don't even know if there's a function for that, except iridescence is a structural thing rather than pigments, and the structural part of that membrane might strengthen it and help to waterproof the spores while they're developing, and just keeping it free from invertebrates.
Oh, Sarah, just wanted to pick up on a point. You're talking about amoeba, and then you have a male and a female. That's part of the life cycle, is it?
Yes, yes. So
Right.
two compatible amoeba mate,
Oh, right. Oh, thank you.
and that's how it's usual sort of thing.
I'm intrigued by the name Plasmodium because I know Plasmodium as malaria.
Yes, yeah.
Any connection?
No, absolutely none. I don't know why it's called Plasmodium. I was thinking of that on the way in. I thought, malaria. Yeah.
Are they all syncytial like that, or are they
all-
some of them cellular?
No, they're all acellular. So the cellular slime moulds are a completely different group of organisms. The ones that I'm studying are the acellular slime moulds, called the myxomycetes. The cellular slime moulds are called dictyostellids, and they have there's another single cell organism. The cells live in the soil, and they actually have a slightly different way of getting together. They emit a substance, and as a result of that, the different cells come together. They form a structure, a fruiting body, with only one or two spores, but they're always found in the soil. You don't see them with the naked eye.
You mentioned citizen scientists, and I presume you include yourself in that category. And I'm just wondering, and you obviously have a lot of connections with academics around the world. How does I find that interesting, citizen science working with professional academics. Could you go on a little bit of how you found that?
I suppose the association with the academics came about through my association with Fungi Mat, and just getting to know the different academics who study the different groups. I did have the opportunity to meet some of these people in person. We went to a slime mould conference in Costa Rica in 2019. Is it 2019? No. 20. It was just as that unmentionable virus was making its way across the world, Ron and I were travelling to Costa Rica. It was extremely nerve-wracking, but it was really great to meet those researchers. And I think if the virus hadn't have been wreaking havoc across the planet, a few of those researchers would be in Tasmania, or they certainly would. They were certainly intending to visit, so it's a real pity. And we hope they come because they are interested in what's being found in Australia.
Sarah, very interesting talk. I know nothing about slime moulds. Well, I do now, but I didn't. do they survive land clearing and agriculture? And if not, what do we lose out of the soil and out of productivity?
Yeah, it's a good question, isn't it? They're very sensitive to climatic conditions. I imagine if our place was cleared, they would be eliminated. It's hard to know what's going on in the soil. I know there's this real push for regenerative agriculture, and ideally they'd be talking about getting organic matter back into the soil so that these amoeba can proliferate, but I don't imagine there'd be many slime molds left in agricultural soils.
Hi. As a complete and utter novice, where would one even begin to look for slime mould, or what equipment would you need and how would you go about starting?
Well, probably in your backyard if you've got any organic material in your garden. They're very common. They're actually much more common than we ever suspected. This person in Victoria who I've been communicating with, she has been looking just in their school grounds. She's got school students really keen, and she's found slime moulds just in the leaf litter in the school grounds. And I think she's found 50 species within quite a short distance of where she lives in Melbourne. So they are very common, and I know people, they turn up in pot plants. I found them in the compost heap and on raspberry canes and rhubarb stalks. My most important piece of equipment is a hand lens, so a 10 times hand lens. I've got one on, I did not fiddle with that. Whoops. and a very strong light. So when I'm in the forest, I use a really strong headlamp or torch, and that just shine it along the log. But yeah, it takes a while to get your eye in to actually figure out what you're looking at. And some of the very small fungi do resemble slime moulds.
I just have one question. As those cells come together to make a mass and then build their structure around that, are they building a cytoplasm?
Yeah, yes, cytoplasm, yeah.
So, okay, my second question on top of that is, is there a catalyst that triggers that cytoplasm from being formed? Like, is it a mass or is it a time?
I'm not sure about the formation of the plasmodium, but you raise a good point, because the plasmodium actually, it moves by what's known as cytoplasmic streaming. So it'll move forward for about 60 seconds, then it'll stop for about the same time, it'll go back for about 60 seconds, but ultimately it will move forward. So that's all moving the cytoplasm through that plasmodium. And possibly it would be so this is the feeding stage, so it's feeding on bacteria or anything on the surface of the log, or fungi, bacteria, algae. So the shuttle streaming, as it's called, will be to distribute the food through the fruiting of the organism, the plasmodium.
Thank you.
If they don't have a brain, how do they know what to do?
Yeah, that's a good question. Well, you got? We don't really know. They're amazing things. They sense it, and I often get people send me the latest research on slime moulds, and people and they have at the plasmodial stage, as I said before, this is the most studied of all the microorganisms at the plasmodial stage. They do, they can find rolled oats if they make a maze. They can find rolled oats. My reply to that is they've been around probably 600 million years, possibly even 1 billion years, so they certainly know their way around, even if they haven't got a brain.
How do you account for the mysterious appearance and disappearance of them from year to year?
No idea. No, that just keeps me studying them. Yeah, there's no rhyme or reason really. I thought that they would all just appear on the same logs just like fungi do year after year, but they don't. And that's, as I said, that's one of the advantages of living where I do, because one of these tubiferas that I found and was described in the new species, I haven't seen it since that year. So if I hadn't have been that year, it would have been still mysterious.
Maybe following the springtails around would help.
If I follow the springtails?
No,
The Plasmodium?
follow the springtails around because they seem to be quite numerous.
Can you either repeat the question for us or get repeated with the mic?
It's about springtails, you know how they jump around in the water.
Yeah.
Well, I just wonder whether the slime follows them, because they can be quite numerous in some places.
No, I don't think so, because the slime's not really interested. It's more the springtails are interested in eating the slime rather than the other way around. So I don't think they'll be doing that. Yeah.
Well, thank you for a most delightful talk. We've been looking forward to this because we know your beautiful pictures.
Thank you.
And now we know what we're looking at. It's our practice to present you with a certificate to remind you that you spoke to us.
Oh, thank you very much.
And one of the Society's publications, which I hope you haven't already got.
Thank you.
So thank you very much.
Thank you.
Would you join me?